Our first keynote speaker is Dr. Lalit Batra from USA. We are very thankful to Dr. Lalit Batra, who has connected to us on a very short notice and at very odd hours. It's my pleasure to welcome Dr. Lalit Batra from Louisville, Kentucky, USA. Dr. Lalit Batra is postdoctoral research associate from Center of Predictive Medicine, University of Louisville, USA. I'm very proud to say that he is an Indian researcher doing his work at international level on very important subjects like cancer and immunology. He has been working in Louisville since 2050 after qualifying GRF, NET, and also got a scholarship from He has teaching experience also in India, and he is recipient of many awards at national and international level. Like Young Investigator Award and 2 awards in immunology and many fellowships. He has many international publications and has presented papers in many national and international conferences. He has done many specialized training courses on environment, health, and safety by University of Louisville. He is member of 4 national and international bodies. His achievement speaks volumes about how Indians have established themselves in advanced researches and brought laurels globally. Students, ये हमारे लिए मोटिवेशन है कि हम किस तरह ग्लोबली भी काम कर सकते हैं. Think locally and act globally. Today, he will be sharing with us his expert talk on GRFT, a broad spectrum antiviral protein. Now, I welcome Dr. Lalit Batra. Welcome, sir.
Hello, everyone. Thank you, ma'am, for your words. Thank you. Thank you very much. Let me start with the screen share. Can you see my screen?
Yes, sir. Carry on. Carry on, sir.
Thank you very much. Thank you. Thank you again, ma'am, for your words, for my introduction. Let me introduce myself to everybody again. So, myself, Lalit Batra, I did my master's in biotechnology from BHU Varanasi, and then I did my PhD from DRD Gwalior, that is the Defense Research and Development Establishment. There I worked on the plague vaccine research. Then in 2015, I came here in United States for postdoctoral associate position. And here I'm working in a lot of projects like cancer, diabetes, immunology, and recently COVID-19. So let me start with my first project. So my first project was on the cancer immunoprevention with SA4-1BB ligand. So SA4-1BB ligand molecule is an important regulator for the 4-1BB ligand pathway. This 4-1BB ligand is an important regulator for the Th1 immune response. It activates a lot of innate immune and activate the other component of the system. So here we generated a novel form of the 4-1BB ligand. It's a SA4-1BB ligand. So we conjugated the streptavidin with this 4-1BB ligand. So with this, it forms a good complex and it has a tendency to form oligomer and tetramer, and it can be, you can load any antigen on that and it will be delivered to the targeted antigen site. So initially in our lab, before they work on this adjuvant. This is SA4-1BB ligand adjuvant, and they use for the treatment for the HPV and other cancer antigen, and they treat the mice with this as an adjuvant. Later on, when I came, I started working with this adjuvant, and then I observed that with this treatment alone with this adjuvant, there is no need of any cancer antigen. So we can give it to the mice, and the mice, when we give this immunize with this SA4-1BB ligand, and then immunize the mice, and then give the tumor, the mice they were tumor free. So first of all,
Participants, please mute yourself. Again requesting, please mute yourself. Participants.
वो आ गया ना सोडियम वाला, तब से मतलब अलग टेस्ट है इनका बिल्कुल। हाँ
C6F10 melanoma to the mice, and the mice they were tumor free after the tumor cell
इसके बदले
20 melanoma and the 41 breast cancer cell line. So the most importantly, this immunopreventive efficacy of 4-1BB ligand, it starts from the 2 weeks and it is there until of the 14 weeks. As you can see from the graph, so the efficacy started from the 2 weeks, 4 weeks, 8 weeks, and it remained until the 14 weeks. And later on, then we characterize why we need this as a prevention agent. So this is important for the patients that have the hereditary to some mutation, they have P53 mutation or the preneoplastic lesions, or they are or the person working in the smoke cigarette industry, which are more vulnerable to cancer. So this molecule can be given to them, and they can be cancer free. So, and later on we decipher the mechanism. Later on we decipher the mechanism behind this prevention, and we found that CD4, CD44, NK cells, they are playing role, and the CD4 and a lot of CD4, CD44 cells are interferon positive, NK cells are interferon positive, and NKT cells are interferon positive, as you can see from the significant bars. So this research was published in the Cancer Research in 2019, and part of this research has been in communication with the Nature Medicine journal. So this is my first project on cancer. Now my other project was on the diabetes. So in the diabetes, we are using on the islet transplantation model. So the people, there is a new approach. The people who are type 1 diabetic, they can be given to the islet so that the beta cells, they are functional. So they can be treated with this islet transplantation. But this islet transplantation has some problem because the people has to be on the donated islets, but there is a like allogenic response. If somebody else can, he will give you its tissues or one, there will be a lot of allogenic response, and you have to be survived on a lot of immunosuppressive drug. So we want to cut off this immunosuppressive drug regimen. So here I use the immunosuppressive molecule, that is a PD-L1. So similarly, we ligated with the streptavidin, performance, oligomer and tetramer, and then when you have like biodynamic cells like islets or splenocyte, you can very well add on to them, and they are being nicely decorated. So these are the bindings that are added with the SAPDL1 molecules. So this is our mechanism is our experimental how we perform the experiment. So they are the P6 mice. First we give them streptozotocin. It makes the mice diabetic. That is, it kills the beta cells of islets. And then these mice are diabetic within like 4 days. Here we have isolated the islets from the other mice like bulb C. So they are variety are allogenic. B6 and bulb C, they are allogenic mice. So we harvested the islet from this and decorated the PD-L1 protein onto them and see how the graft are surviving. So to our surprise, when we give the SAPD-L1 only, the mice survive expect to control up to 40 days. But this treatment was not effective. But when we give the SAPD-L1 immunosuppressive drug, rapamycin, not for a long time like for the generation we have to give for like short course of 15 days. The mice, they were diabetic free until the end of experiment, that is the 100 days. So with this notation, this molecule is under the it is non-human under the non-human primates. Now this experiment was on mice. Now this regimen we are applying for the non-human primates like monkey, so that a short treatment of rapamycin suppress the allogenic response, and we can target the diabetic, we can overcome the diabetes. For the mechanism, we see that with this treatment, we are seeing a lot of regulatory cytokines like FOXP3, TGF-beta, and a lot of anti-inflammatory cytokines like IL-10, IL-4. On the other hand, we see a increase in the and decrease in the pro-inflammatory cytokines. In the red bars, you can see the red dots, a decrease in the pro-inflammatory cytokines like interferon gamma, TNF-alpha, and IL-1 beta. When we at the end of experiment at 100 days, we harvested the kidney from these mice, and we see a lot of CD4 cells with FOXP3 positive. That means these cells are the regulatory cells. So they are playing the roles, CD4 FOXP3 cells for their mechanism. So this research has been published this year in Journal of Immunology, Science Advances, American Journal of Transplantation, in two journals. So these are like separate part of what we publish in different journals. So the Science Advances is have for the non-human primates. We publish that for non-human primates. So this is my second project. My third project was on the infectious disease. So in India, I work on the plague vaccine. Here I got a chance again to work on the plague vaccine. As I told you earlier, this SA4-1BB ligand molecule, I work with SA4-1BB ligand molecule for the plague vaccine. Plague, everybody knows that plague is caused by first Yersinia pestis. Like we are seeing this COVID-19 pandemic, there was like a long back ago, there was a pandemic for the Y pestis. It's called plague. It killed a lot of millions of population way back in 1500 or something like that. So in India, the people in the DRD, they are working for the development of vaccine against this plague, deadly bacteria plague. Here, the people, they are also working on the development of vaccine against plague. So here, for the plague vaccine, what we did, we have the F1 and B antigen of Y pestis, and we have given these mice, the mice to the F1B with SA4-1BB ligand protein. So this is a like short brief experiment. So here we generated 3 groups. So in which we give the F1B with alhydrogel and boosted again with this regimen, and then we give the F1B with alhydrogel and then boosted with F1B with 4-1BB ligand. In third group, we give the F1B with alhydrogel and 4-1BB ligand, F1B with alhydrogel and we give 4-1BB ligand our adjuvant. So as you can see, the third group has maximum amount of interferon gamma 0.186 from the CD44 cells and 1.53 as compared to other groups, 1.53% of interferon gamma coming from CD8, CD44 cells. So as you can see in the form of absolute cell number, we see a lot of a very significant amount of interferon from CD4 interferon from the last group, that is F1B with alhydrogel and 4-1BB ligand with the same boosted dose. Similarly, we see the CD8 interferon gamma T cells. Here we see a lot of IgG2C coming from the last group, that is alhydrogel plus 4-1BB ligand. So when we give the mice the challenge with like 10-80-50 to the mice, to the immunized mice, we see a like reduction in the survival, but we didn't achieve the complete protection. Then later on, we are standardizing the doses of the vaccine, and we are getting the complete protection. This research has been published in Vaccine in 2019, and a part of the work is that we see the complete protection is again communicated in the vaccine journal as a part 2. So as of my interest in my PhD and in the postdoc for the infectious disease research, I came into the play for COVID-19. So I started working for the COVID-19 therapeutics. So for the COVID-19, we are working for the therapeutics for COVID-19. We are utilizing a lot of antiviral and other vaccine approach for the prevention for the COVID-19. So here we evaluated a novel molecule called Griffithsin. This is a broad spectrum antiviral. This Griffithsin is a natural product for the red alga Griffithsia. It produces a lot of Griffithsin from its leaves. So this Griffithsin we synthesized in the molecular biology lab. This is a 121 amino acid, like 12 kDa protein. It has a carbohydrate binding motif, mainly the mannose binding motif. So it effectively binds with the virus and on the envelope virus, not on the non-envelope virus, mainly on the envelope virus like HIV, HCV, MERS, and SARS coronavirus. When it binds to the virus with their envelope protein, the virus, it can't bind to the cell surface. So it prevents the entry of virus to the host cell. This Griffithsin molecule is a very stable, it is heat resistant, pH resistant, and it has no observed toxicity on the in vitro and in vivo on the host cells. So we are utilizing these molecules for the COVID-19 pandemic therapeutics. So earlier our lab has has shown a beautiful result of this Griffithsin. We call it as QGRFT because this is modified from the GRFT. It has another amino acid modification. So our lab has shown this QGRFT therapeutics against SARS corona and MERS coronavirus. They are they all SARS-CoV, MERS-CoV, SARS-CoV-2. They belong to the family coronavirus. So earlier they tested this molecule on MERS coronavirus and SARS coronavirus. When tested on the MERS coronavirus in 2016, they published the result in 2016 my lab. So they used the 3 cell lines HUH7, MRC5, and Viro81. This is the control, non-infected. Other cells they are the infected, but are treated with different doses of Griffithsin 0, 0.125, 0.25, 0.51, and 2 microgram per ml. As you see, you can see a lot of virus, MERS coronavirus, that is red. It is the spike protein of MERS coronavirus. You see, without 0 means there is no Griffithsin. You can see a lot of virus in these cells, but as you see, like higher doses, this virus clears off. So this is the titration graph, how they titrated on the HUH7, MRC5, and Viro81.7, and with the increasing dose, they see a dose-dependent inhibition of the infected cells with around like 0.5 EC50 for the HUH7, 0.5 for the EC50 for the MRC5, and 0.25 or like 0.5 EC50 for the Viro81 cells. So they showed that this Griffithsin molecule is effective against MERS coronavirus. That is the same way, like it is the same as like SARS-CoV-2. MERS has the spike protein. So this, so they predicted that this molecule it can be effective against SARS coronavirus. So later on they tested the SARS coronavirus, and they shown in vitro as well as in vivo. But MERS coronavirus, they have result for the in vitro. SARS coronavirus, they also show a dose-dependent decrease in the inhibition of the virus, and the cells are inhibited from the virus infection. Then they ran the mice with Griffithsin and concurrent treatment with SARS, and they observed that the treatment, after the treatment, the animals were virus-free. They don't have any symptom for virus. They didn't die, and the mice they were survived. So this treatment showed that Griffithsin were against SARS coronavirus. So now the papers have published from 5 years that is Griffithsin is effective against human coronavirus, human immunodeficiency virus, that is HIV, Nipah virus, and others. So these are like I will say this Griffithsin is a broad spectrum antiviral inhibitor. Now I will show you the SARS coronavirus result. These results are confidential. So this is our first experiment on the Viro cells, that is in vitro experiment on the Viro E6 cells. So we have the cells, Viro E6 cells, we give the virus as well as the different doses, started from 0.312 microgram per ml until 20 microgram per ml. We mix this protein with the virus so that the virus can bind to protein, and when we give to the cells immediately, like mixing within minutes and then give the cell, it should not bind to the cell. But you can see like any treatment is look like 100% effective. So we see a like very good efficacy at around like 5 and 10 microgram per ml. These are the cell control, these are virus control, and these are the treatment. As you can see, there is a lot of cytopathic effect on this Viro cell. They are only infected with the virus. You can see a lot of cells they are dying because of the virus infection. But here in these 2.3 and 0.6 to 5, some of the cell, a lot of cell died in this 1.25 microgram per ml treatment. Some of the cell died, but here the increasing concentration, the death of the cell decrease in the dose-dependent manner. So this is the graph that we generated from this data, like how much dose of Griffithsin we need to kill to protect our like 50% cell from the virus infection. So amount of effective concentration of Griffithsin come around like 1.31 microgram per ml, and this will protect like half of the cell from the virus infection. Now we expand this data to the human tissue. So we have this is like another experiment. So as we all know, the SARS-CoV-2 it infects the human cell with ACE2 receptor. So what we did in our lab, we have the Viro cells. So Viro has ACE2 positive as well as ACE2 negative. So we sorted the cell with the flow cytometry sorting machine ACE2 positive Viro cells, and then we used these cells to treat the cell, to infect the cell, so that there like prevents infection of the cells and all of the cells should die within a few MY. So when we have this ACE2 positive Viro and virus infection, we propagate this virus in the ACE2 positive Viro cells, so that we should have a lot of PFU titer and we can titrate a lot of antiviral and other drugs on these ACE2 positive Viro cells. So this is our next regimen. So this is a cycle. So first we have to use the in vitro and then in between model and then animal model. So this is the in between model, the human three-dimensional tissue. We use the epi airway and epinasal tissue for the Griffithsin prevention efficacy. So we treated so we have the epi airway and epinasal tissue. We treated the concurrent treatment, like combined treatment with the virus as well as the Griffithsin. We mix both of them and then we give to the epi airway and the epinasal tissue. So this is the cell control. You can see there is nothing virus titer in the, but in the MO 0.1 MI there is like 10 to the power 8. So this is, so we give the virus and then we collected the superattend at different hours to see the virus progression and our progression, how we are preventing with the GRFT. So these are like, you can see at 120 hour, you can see at 0.1 MI there is a lot of virus, 10 to the power 8 CTRBEPR 50 per ml in the virus control, but in the this is our Griffithsin treatment tissue. So we have chosen different dose like 67 microgram per ml, 6.7, 0.67, 0.067 ml Griffithsin. So as you can see, as compared to control of any at hour, like we can at 72 or 96, 72 or 96 or 120 hour in the control tissue there is around like 10 to 8 or 10 to 6. CID, but in our treatment there is around like 10 to the power 2 or 10 to the power 3 CID 50 per ml. We see a very good result with the epi airway with Griffithsin treatment, and then similarly we have the result with the epi airway tissue. So they are with the same manner, and but the efficacy was not good in epi airway tissue. So we have this efficacy, very good efficacy in epi airway tissue. Upon at the end of experiment, we need to see what is the mechanism behind this Griffithsin protection against SARS-CoV-2 infection. So we did the whole RNA sequencing of this their tissue, and we observed that. So this is the RNA sequencing data of SARS-CoV-2 infected and GRFT treated human epi airway tissue, because we see a lot of good virus inhibition in the epi airway tissue. So when we compare the Griffithsin versus virus control, we see a lot of chemokine and cytokine. A lot of cytokine, they are membrane associated, like chitinase like 3, paralipin 2, platelet growth factor. But we see some of the cytokines that can be immunoregulator like IL-33, DEPP1, ST3, GL4. Looking at the global picture, we see, I can say that there are a lot of cellular metabolic pathways that are involved with Griffithsin, but there are two pathways, that is hypoxia-inducible factor 1 and IL-17 signaling pathway. So they are playing them role. So in the immune response, there are three types of response. I will say now three types of response: TH1 response, that is the pro-inflammatory response; TH2 response, anti-inflammatory response; and TH17 response, that is the IL-17. This is a balanced response between TH1 and TH2. This TH17 it regulates TH1 as well as TH2 depending on the condition. So like SARS-CoV, in the COVID infected patient, there is a lot of pro-inflammatory cytokines like interferon, TNF-alpha. So if this GRFT induced TH17 signaling pathway, then it will go to the system and then in the immune system, and then it will regulate the TH2 pathway, so that there should not a cytokine shown as we see in the COVID-19 patient. So this is the very good data for the experiment that this GRFT is effective against SARS-CoV-2 infection. Now we are our next target. The leftover is the test that this Griffithsin molecule against MERS coronavirus infection. And we have like, as you all know this, we don't have any model, well-established model for the mice model for the COVID-19. We are establishing ferret, hamster, as well as EC2 positive mice. That is they are EC2 mice, EC2 positive mice. They are humanized. They are transfected with humanized EC2 receptor, so that they should be susceptible to the COVID-19. So our next target will be to utilize the efficacy of Griffithsin against is in SARS-CoV-2 infection in EC2 mice, ferret, and hamster, and we have a good data coming for EC2 mice. So with all this, I will say thank you. Thank you, Apna Madam. Thank you, Dr. Pratima Srivastava, for giving me an opportunity to present our at the CTRBEPR and JDB College. Thank you very much. These are some of my publications from here for cancer, diabetes, and infectious disease. If you have any question, please let me know in comment section, or you can have my email ID. You can ask any question. So there is like a lot of research opportunity for the people that are interested in virus infection, and because now in United States a lot of people they are working on the virus infection, and there is a lot of funding coming from the Democrat that they ask for the work on the infectious disease. So now if you have the student, they have calibration and interest for the vaccine research, for antiviral, or any kind of virus research, they can come and they can work. Initially, they have to just only they have to publish 2 or 3 good papers. After that, they will get a very good postdoc position here, and then they can work in that to their field or thank you very much.
Thank you, sir, for your very nice deliberation. Now, the participants, if you have any query, you write your queries in the chat box, and we will connect with Batra sir, and you will get satisfied answers thereafter. Sir, I want to know one thing from you, from my side. In the present scenario, can you suggest us some measures we should take to protect ourselves from this COVID-19 or the corona?
You are asking like preventive measure, like physical preventive measure, or like medicinal?
Yeah, medicinal side.
Medicinal side, yeah. Physical, because physical, everybody knows, use mask, keep distance from others, 6 feet distance. So like medicinal, I will say like this, I will say like Griffithsin, I will talk in like natural manner. Like Griffithsin, this is a natural product. So I will say use natural product as much as we can possible. There is a lot of Indian medicine that we can use for this, so prevent the infection.
So you also suggest that what Indian kadhas and things we are from plant resources we are using, like Keynote and all.
Dr. Pratima a very good remediation, it's failed in the clinical trial. So now they back up the remediation. So the remediation was a great drug for the COVID-19, but it's better. There is now no in the US, nobody should treat with remediation. So I will say use natural product, or like whatever you know in the traditional culture or in the ancient culture, because everything lies in our Indian culture.
Participants, I again request you, if you have any queries, write in the chat box. Over to you, Alpna.
Very nice presentation, sir. Thank you, Dr. Batra, for giving us information on antiviral protein in present scenario of COVID-19.